Okay, so we've talked a lot about DNA, and I mentioned that the order of the bases in your DNA sequence is what makes you you. But we've kind of missed the part of how having this sequence of A's and T's and G's and C's means anything to the instructions to make you. So this is where protein synthesis comes in, where we take that DNA sequence and we turn it into something usable. Those are called proteins. So protein synthesis, we're going to write here, is the process of turning DNA into proteins. But, of course, it can't be that easy, so there has to be an in-between step, and that's called RNA, so by way of RNA. And to help you remember, RNA is like DNA, but it's single-stranded. And also, it uses a different base. It uses uracil instead of thymine. Okay, so to remind yourself, let's make a little flow chart over here. We're going to say it's DNA makes RNA makes protein. Okay, so we're going to talk about this in terms of our cute little firefly friend here. How does a firefly glow? Coincidentally, it takes DNA to code for the protein, which causes a firefly to glow. So let's figure that out. We're going to go over here to the green start. You can color yours whatever color you want. Green's my favorite color, so it just felt like a start idea. So you're going to see here we've got our DNA inside our nucleus. So the DNA is trapped inside the nucleus because it's too big. So DNA cannot leave the nucleus. That's really important and why we have RNA. The rest of this process happens out in the cytoplasm of the cell because that DNA can't leave. So the DNA can't leave, it needs to have RNA. And we can tell this is RNA because it only has the one strand. So DNA and RNA don't speak the same language. As I said, it doesn't have the base thymine, it has uracil. So we're going to start with step number 1, which is called transcription. So think of this as DNA giving instructions to RNA. The RNA writes it all down, is going to take it outside of that nucleus. So a nucleotide, remember that's the A's and T's and G's and C's, or in this case U now. So a nucleotide sequence is copied, is transcribed from DNA to RNA. Okay, and this piece of RNA is called messenger RNA. But we like to shorten everything, so messenger RNA is referred to as mRNA. So this messenger RNA is the RNA that carries information from DNA to the ribosome. So the ribosome is what takes this instructions and turns it into a usable product. So think of it like blueprints. The messenger RNA is taking the blueprints from the original shop, the DNA, taking it to the ribosome, saying, here you go, this is what we need to build. But it has to be translated first because the body can't read what's in the RNA. So we have to go through translation. So the mRNA sequence is decoded. So it has to be turned into something that the body understands, and it specifies the amino acid that needs to be built. of a specific polypeptide. So those polypeptide chains are what makes up the amino acid, and those polypeptide chains are made up of specific amino acids. So let's go look at decoding. So the ribosome here has 3 amino acid, or 3 bases in it, because it reads them 3 at a time. And that's called a codon. So a codon is made up of 3 bases. And they correspond to a specific amino acid. Or, so they can correspond to a specific amino acid, or they may also tell the ribosome to stop or start. So the entire DNA sequence is not being transcribed at the same time. It's told where to start and where to stop because it goes gene by gene, because not all genes need to be turned on at the same time or at the same point in your life. Maybe some are only when you're younger, some are only when you're older, some are only at certain times of the day or when under certain stimulus. So there has to be starts and stops, otherwise the whole thing would just go crazy. And we find this out by looking at these codons on a codon wheel, which we'll look at in a second. But first we're going to practice going from DNA to mRNA. So this is our transcription. So we're just going to go base by base and turn it into the mRNA sequence. Remember, if we know what one base is, we can always figure out what the opposite strand is. So let's pick a new color. Pick green, because green's my favorite color. So when we have a T, T's go to A. Remember, apples and trees, cars and garage. But there's no trees anymore. There's U's. So A goes to U. C becomes G. We have C, U, G, G, U, A, U, C, my favorite, C, 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 A, U, G, G, U, C, and U, A, G. So that's transcription. We've turned it into the code that the mRNA can get out of the nucleus. But that's great, but that doesn't build anything in the body. We need to translate it into amino acids. So this is where we have to go to our codon chart. So I like to use the codon wheel because I think it's easier to understand. Okay, so if we go here, let's look at our codon chart. So the first one, if you look at your mRNA, says AUG. So how we do this is we find A right here in the center. We have A. And then we have to find our second one, which is U, right down here. And lastly, we see the G. And this tells me that the first one codes for methionine. Now this chart is not super helpful at this point because it doesn't tell us that when we see it in the beginning, AUG also means start. And I like to put lines between them because it reads it in a frame of 3, and that way you don't get confused, because if you get off by one, you can completely change the course of the amino acids, which does happen in the body with issues with mutations. Okay, so the next one says CUG. So we're coming back over here. We find C. We have C, we have U, and we see that when we have C and U, no matter what your third base is, it always codes for the same amino acid. So we have leucine. So this codon chart gives you the 3-letter abbreviation. Some will give you a 1-letter abbreviation, but they do have longer names. So let's do one more, GUA. Okay, let's go back here. So we have G, we have U, and no matter what your third base is, it gives you valine. So let's write that, VAL. Okay, so you're going to continue with this sequence using your codon wheel or codon chart. Okay, so if we go up to our ribosome, we see that the ribosome here is reading these three bases and calling for an amino acid. That amino acid is brought over by what's called the tRNA, and the T stands for transfer. It's grabbing those amino acids and bringing them in, so the tRNA. And it carries the amino acid to the ribosome. So you'll notice that what the tRNA is bringing in is the opposite bases for what the ribosome is calling for because the way they fit together. Okay, so our ribosome has our tRNA bringing in the amino acids and it's spitting out this amino acid chain. So here's our polypeptide chain. So if we look over here, the last step is that the amino acids are linked in that polypeptide chain. And the polypeptide chain is held together with peptide bonds. So now if we go up here and look at our firefly, he's got his sequence read, translated into amino acids. The amino acids are bonded with the peptide bonds, and then they're folded in a specific way because the fold is really important to how it interacts with the body. And now our firefly can glow. So the protein here codes for an enzyme, and that enzyme is called luciferase. Now I've mentioned a key piece and left this part down here blank. I said the fold is important to how it interacts in our body. So when that fold doesn't work right, we get something called a prion disease. So it's all in the fold of the protein. That's really important. So how it folds relates to its function. And if it does not fold correctly, we get some problems. And these are called prion diseases. So a misfolded protein, the prion, causes things like spongy brain, or if you've heard of mad cow disease. And the reason those are so bad is because a misfolded protein, that prion, can cause other proteins nearby to also misfold. So consuming that mad cow disease meat could trigger your proteins to also misfold because they send out a signal. There's another cool one, I use that term loosely, that causes insomnia, where the people stop sleeping for days and days and days until you eventually die because your body needs to sleep. They're both simultaneously amazing and horrifying at the same time. Okay, so we're going to write that a misfolded protein called a prion can cause disorders such as BSE. That's what I was thinking. BSE, which is bovine spongiform encephalopathy, mad cow disease. Prions cause other proteins nearby to misfold. Okay, so that is protein synthesis, and we've figured out today how our firefly can glow.